Hello everyone. So today we are going to learn about the multiple sequence alignment. So basically multiple sequence alignment is a sequence alignment of three or more than three sequences. It can be done for protein sequences, for DNA or RNA sequence. So basically we today we are going to perform the multiple sequence analysis, which is highly preferred if we are going to search for homology, like if they are having an ancestral relationship among the sequence, or if we have to find like how the insertion, where is the deletion is being done in the sequence. So it is a dynamic programming. I will demonstrate one and two tools so that we can learn more about the multiple sequence alignment. Here you can see there are multiple nucleotide sequences. Basically it is a protein sequence given here, and these are some colorful patterns, and these are multiple sequences which are being like compared in this diagram, which is present on Wikipedia, so you can go and check also there. So basically, multiple sequence alignment plays a highly important role. There are multiple types of algorithm alignment methods are there. There is a dynamic programming method, which is like optimal alignment is done so that the gap penalties, the substitution matrix is made. So these are different types of alignment matrices which are given. They also go for progressive alignment construction, in which with a similar like in initially we have a similar sequence, and in the last a distant sequences can be grouped and compared. So it is a relationship in which like different sequences as they are depicted as a tree or a guide tree is used, and then adding sequence sequentially to the growing multiple sequence alignment according to the guiding tree. So these are multiple sequences. You can also compare a small sequence, a stretch, or we can go for a complete sequence comparison. So it is highly used to create the phylogenetic tree. If you want to confirm the evolutionary analysis or the hidden motif among the sequence, to find the functional domain, it is also used to find the evolutionary relationship, as I said earlier, and important sites. Like sometimes we have a DNA binding or a protein binding domain. So it is consensus sequence. For finding that consensus sequence, we have to go for like multiple sequence alignment has to be done over there. So there are a lot of different tools which are present to perform multiple sequence alignment. We can like say MAFT tool is there. I guess here are some tools written. If not, I will just, you can write the names, I can speak the names. So we can go for H align T coffee method. We have Clustal W, Clustal Omega, Prank, Mulan, FSA. Base by base is also done, which is an iterative algorithm based method. MAFT is a quick large DNA is compared using MAFT, and it is a progressive alignment method. So today we are going to learn the method which is Clustal Omega alignment method. So first of all, it is a progressive method which will be comparing one by one. Each residue will move one by one. So first of all, what you have to do is like go on the portal. Here you can type what Clustal Omega or Clustal W. So I am going for Clustal W. Here is the tool. We can click on this link, and for this, one second. So here in this tab, you can write NCBI because we are going to take the sequences from the database. It can be any database. It can be EMBL. I have demonstrated all the databases according to the requirement in my previous videos. You can go and watch them there. So I guess I will be taking, we can take any sequence. So we are going for protein sequence. So you can click on protein and you can type any protein. I am interested in insulin. So just press on click and it will open sequences. So for demonstration, we can open the faster sequence. 2 to 3 faster sequences we can take. It is a shortcut method. Otherwise, you can go open this file and then you can go and click on faster sequence. I have opened the multiple tabs over here, so it is easy to see them. So what I am going to do is like copy the whole, like accession ID should also be there. And I have to do what? I have to paste that sequence here and then clicking on the next tab and next line, I will go for the another sequence. Like you can type also, or you can give the file also. But yes, you can give the file option is also given here. So you can also like download and upload the file, or it is an option like for a good option is like you have multiple sequences, so you can go and copy them in a notepad and then you can upload that file here. So it will be an easy method if you have multiple files or multiple sequences you are considering. So I have pasted all the 3 sequences over here. Now you can see it is a protein sequence. So I have selected the protein. I can also selected the DNA if I was going for it. The results operation are like PIR format, FASTA file format, or cluster format. So you can go and according to it, you can work. MAFT is also multiple sequence alignment tool. So in this video, I am going for Clustal W. So you can also find like here is an fast and approximate, because when the things or an algorithm is working quickly, so the approximation like alignment will be done. But if we go for slower, so it will be accurate. As we know, the slowly it will move, the more accurate results will be there. But yes, for the demonstration, we can go for approximate results, which will be fast and easily will be present on our screen. But yes, after that, you can try yourself for the slow one. Then you can see here, like moving downwards, here is a window size. As I previously said, like window matching is there. In my videos, I have demonstrated like how the window matching is done one by one progressively. The movement occurs. Gap penalties, how much gaps will be sequence and like how much top diagonals will be there. A diagonal whole matrix is study like PAM blossom matrix, so they have to be created in a such a way so that these all things matters over there. So you can also select the matrix according to go for PAM matrix, blossom matrix. Multiple matrices are there with a weighted matrices, and according to the weight, the evolutionary relationship and the linkage between the sequences is being calculated. So I will be going to execute the multiple alignment now. So it has like calculated the result. Yes. Now you can see there are some dashes like where there is a gap in the sequence and the sequence was not able to match with the other sequence. Now you can also see like these stars which are designating the exactly matched nucleotides or amino acids. In this case, we are considering protein. So it is amino acid which are exactly matched among the sequence. Here you can see in all the 3 sequences we consider, this position is L, L and L. So it is highly conserved. And this like colon is there when it is maximal matched. It can be like amino acids are also there, like hydrophilic to hydrophilic, hydrophobic to hydrophobic, basic to basic, acidic to acidic. So these all are being checked and we can find like where is the conserved motif. So I can see from this LCG is a conserved motif which is present here. But yes, if we consider the alignment is not good, that is okay. But there is no evolutionary relationships. You can also see the amino acid size also. You can also score, like according to the weighted matrix, the score is also given to the each sequence which we have considered. So hope so this video was useful to you. For more videos, please subscribe this channel. Thank you.